I'm preaching to the choir here about induction of phage, and so in that context, I realize that many of you know more about the induction than I do. Basically, the take-home message that I'm going to go ahead and let you know is, as Forrest indicated yesterday morning, that we probably need more dialogue on, specifically on what antibiotics are doing to bacteria in inducing prophage, along with other consequences that we see due to antibiotic exposure. So I also wanted to go ahead and acknowledge my collaborator on this project was Brian Brunel at the National Animal Disease Center. Our technicians in our laboratories are Kelly Winter and Brini Atkinson. This particular research was an offshoot of a collaboration that I had with several investigators, Heather Allen, Brian, Insoo Lee, Sherwood Kajans, and Thad Stanton, where we showed that the agricultural antibiotic Carbadox was able to induce phage-mediated gene transfer in Salmonella. So my presentation outline is I'm going to talk a little bit about the epidemiology of Salmonella, the prevalence of Salmonella in animal production, and then our investigation of antibiotic induction of prophage in multi-drug resistant Salmonella. So I consider Salmonella an opportunistic pathogen. It's a facultative intracellular bacteria. It tends to be ubiquitous in nature. It can be found in almost all vertebrate hosts, from reptiles to birds to mammals. There are over 2,500 known Salmonella cerevars. Some of these have a broad host range, such as Typhimurium, but others are restricted to certain animal hosts, such as Typhi might be. This is an info sheet from the CDC, and it indicates that there are about 1.2 million cases of Salmonella infections each year, with a medical cost of about $365 million. Of these 1.2 million infections, about 10% of those are due to drug-resistant Salmonella infections. The CDC considers this a serious threat, in part due to the development of antibiotic resistance. And one of these antibiotics that is developing resistance is ciprofloxacin. And currently, I think the level is about 3% of the isolates that they see in humans are ciprofloxacin resistant. But ciprofloxacin is also a very important drug for severe Salmonella cases, but also for other bacterial infections. In animal production, we know that in swine, over 50% of swine production farms can be positive for Salmonella. In dairy operations, you can see 40%, and in about 9% of beef cow operations are positive for Salmonella. We also know that poultry can be routinely colonized by Salmonella also. In most of these instances, Salmonella is colonizing these animals without causing disease. And so essentially the farmers don't even know the Salmonella is present there until all of a sudden they do manifest in disease following things like stress. So in an animal production environment, there's an animal health aspect for the animal. One of the things we're also concerned with at USDA is food safety, but then there's also environmental dissemination. So in animal production, you can have both the therapeutic and the subtherapeutic use of antibiotics. The use of antibiotics may have unintended consequences, and we've been talking for decades about the selection for antibiotic resistance. But there are also other consequences that can occur, such as shifts in the host microbiota. You can have increased toxin production. There could be increases in virulence gene expression, and also the stimulation for horizontal gene transfer. We started looking at Carbadox, which is used in the starter phase of swine production. and it's used for performance enhancement and a control of enteric diseases. Of course, salmonella is considered one of these enteric diseases. And we showed that we could use carbadox to induce prophagia present in salmonella. And so it's your typical lysis curve. We induced with carbadox at OD 600 0.2. About 2 hours later, we saw the lysis. We also marked one of the prophages that is present in Salmonella LT2. That's FELS1, and showed that we could transduce the prophage from one Salmonella strain to another. When we look at a comparison between LT2 and some of the multi-drug resistant Salmonella DT104, we see that there are a few prophages that are present in both LT2 and Salmonella, such as GIFC2 here, and part of GIFC1 is also present. But there are several prophage in DT104, this multi-drug resistant strain, that are not present in LT2. And one of these is the P1 prophage, which is a P22-like generalized transducing phage. So we were interested in whether multi-drug resistant salmonella that contain P22-like phages might be able to transduce non-phage DNA to a recipient bacteria. So we've done this experiment in a couple different ways. One is that we've taken, we have a recipient strain that is His minus and it's kanamycin sensitive. And then we can take a donor strains and we can either use the His genes or kanamycin resistant marker on a plasmid, induce with antibiotics to get our phage production, and then do transductions into the recipient strain. And either select for transduction on minimal media, so that we're looking for integration of the histidine operon here, or we're looking for incorporation of kanamycin-resistant plasmid on a kanamycin medium. So this was a table from our manuscript, and we looked at several of the routine Salmonella typhimurium strains that are used in the salmonella literature, LT2, 14028S, UK1, SL1344. And for those, we didn't expect them to have generalized transduction, and they did not. Then we looked at a series of multi-drug resistant strains, DT104, DT120, DT193, DT208, and U302. And what we found is a subset of those multi-drug resistant Salmonella typhimurium strains did indeed have generalized transduction. So for the fluoroquinolone studies, we're just going to focus on the DT104 and the DT120, since they're able to do generalized transduction. Sherwood-Kazins demonstrated that there are, based on genome sequencing, that there are multiple salmonella serivars that have the capability to do generalized transduction. So therefore, the potential for generalized transduction from many salmonella series is perhaps underappreciated. So we were interested in whether other antibiotics besides carbadox could induce phage-mediated gene transfer from our multi-drug resistant salmonella strains. And what we chose were fluoroquinolone antibiotics. So fluoroquinolone antibiotics have a broad spectrum, and this is kind of a laundry list of bacteria for which the bacteria have susceptibility to fluoroquinolones. And then if you have a particular disease, there's probably fluoroquinolone that might be able to kill the bacteria that's involved in that disease. So the fluoroquinolones inhibit the bacterial DNA gyrase or topoisomerase, and in doing that, obviously they can inhibit they can cause DNA damage, which of course you know will then could induce an SOS response that might induce prophage present in the salmonella. So we looked at 3 different fluoroquinolones, one which is used in human medicine, ciprofloxacin, and then 2 that are used in veterinary medicine, enrofloxacin and danafloxacin. And so we induced with ciprofloxacin, enrofloxacin, or danafloxacin, very similar to what we did in the Carbidox manuscript, where we induced at OD 600 of 0.2, and then allowed lysis to occur, and then did transductions. So the sorry about that. So the 1162 strain is a derivative of DT120 that has a kanamycin-resistant plasmid. The 1165 is a derivative of DT104, and then the 1170 is just a derivative of LT2. It's a negative control. So all 3 of these have the kanamycin-resistant plasmid, and we were able to show that we can get, following fluoroquinolone induction, we can get transfer of this kanamycin-resistant plasmid to a kanamycin-sensitive recipient. We also did some gene expression looking at the REC-A gene involved in the SOS response, and then two genes encoded in the P22-like phage in DT120, and that was ABC2 and KIL. And we saw for all 3 of these genes that both at 60 and 120 minutes, we saw a significant increase in transcription of these genes compared to the 0 time point just prior to the antibiotic addition. So in summary, for this part, the fluoroquinolones used in human veterinary medicine can induce prophage. Generalized transducing prophage are present in some multi-drug resistant salmonella strains. The fluoroquinolone-induced prophage can transfer genes from this multi-drug resistant strain to a kanamycin-sensitive recipient, and the exposure of ciprofloxacin increases transcription of genes within both the SOS and the phage regulons. So I wanted to provide, obviously, some context, and obviously not all salmonella strains have generalized transducing phage, but it has been shown for some salmonella strains that obviously in the prophage they can also encode virulence determinants, and so by using, say, fluoroquinolones, you could also be inducing other prophages that could transfer virulence traits from one salmonella strain to another. Then I also wanted to point out that there have been other reports in the literature for other organisms that fluoroquinolones can induce prophage and or toxins that are encoded in those prophage. And here are 3 examples of that in E. coli. And I'll just highlight this manuscript from Zhang, where they say that these observations suggest that treatment with human S-tech infections with bacteriophage-inducing antibiotics such as fluoroquinolones may have significant adverse clinical consequences, and that fluoroquinolone antibiotics may enhance the movement of virulence factors in vivo. It's been shown in Pseudomonas aeruginosa that ciprofloxacin can induce susceptibility determinants that are prophage encoded. Also Staph aureus, ciprofloxacin can induce bacteriophage. So once again, in summary, antibiotic induction of prophage may enhance bacterial strain evolution, and there are potentially unintended consequences that occur due to antibiotic usage that go beyond just the selection of antimicrobial resistance. And these collateral effects of antibiotics need to be considered when selecting antibiotics for disease treatment. I appreciate your attention.